Thank you for attending this talk titled Modelling Air Conditioning Function During Inhalation. It's great to be a part of the International 2022 conference and allowing us to share this work. My name is Kiao Inthavong from the CFD Research Group at RMIT University, and my collaborators on this project are David Fletcher, Mehrdad Kamushi, Sara Vahaji, and Hannah Salati. Firstly, I'd like to acknowledge the research funding received from the Garnet Pass Rodney Williams Memorial Fund, the Australian Research Council, and from ENT Technologies, Optinose and Nasal for various nasal drug delivery devices. Air conditioning is a dual heat and mass transfer process, and the human nasal cavity achieves this through the interactions between the circulating air in the nasal cavity with the mucosal wall surface, where the latter is supplied with an energy source through a constant blood flow temperature and continual mucus secretion from goblet cells. During inhalation, heat energy is exchanged between the inhaled ambient air and the nasal walls, while mass is exchanged between the water vapor content in the air with the fully saturated mucosal walls. These animations depict the velocity and temperature contour in the mid-plane of the nasal cavity during inhalation, where heat and mass transfer contribute to the overall energy transfer. Cooler ambient air is inhaled, where heat and mass exchange takes place with the mucosal surface. The aim of this work is to expand on existing modelling efforts to account for the dual heat and mass transfer process in the nasal cavity by introducing a new subwall model to account for both the sensible and latent heat energy during the air conditioning process. The nasal submucosal anatomy can be represented with an underlying tissue layer with the blood vessels and capillaries which produce the core body temperature, a mucus and epithelium layer which contain the goblet cells, and the airway region itself. Simplifying the anatomy to a two-layer model, we assume that blood flow properties or blood flow produces a constant temperature defined at the tissue and epithelium interface, and we have a second layer which is the air layer above it. A single equivalent submucous layer treated as a solid material is used to represent the epithelium and mucus region. The nasal cavity region can be represented by the airway and interfaces with the mucus with a coupled thermal condition and a prescribed fully saturated state to represent the wet mucus. The wet surface model was first applied to a pipe geometry to verify its performance and to highlight the influence of latent heat of evaporation. For this test case, the inlet temperature is set to be the same as the wall temperature, suggesting sensible heat transfer is negligible as there is no temperature difference. The relative humidity is set to 0% at the inlet, while the pipe wall is fully saturated. These settings mimic an evaporative cooling process, otherwise known as an adiabatic saturation process. This is where the evaporation occurs from the wet solid surface to the air, occurring at a constant temperature. Heat and mass transfer along the pipe is shown through contours taken at mid-cross-section plane of the pipe and y-planes, which are these circular planes here. The air temperature contours decrease as a result of the latent heat. When the latent heat is excluded, there is no change in the sensible heat transfer as there is no temperature difference. The moisture transfer occurs between the solid-fluid interface, and diffusion into the flow field is evident by the relative humidity gradient occurring in the near wall region. The subwall model was then applied to a human nasal cavity recreated from the CT scans. The two-zonal layer method is demonstrated here with the solid region coloured in red and the air region coloured in white. We note that the solid region is made up of two prism layers, and that represents the submucosal region. 3 steady-state inhalation conditions were evaluated. The inlet temperature was 0.8 degrees Celsius at 20 litres per minute, 10.5 degrees Celsius at 10 litres per minute, and 19.5 degrees Celsius at 5 litres per minute. The top panel shows the mucus air surface interface temperature contour for modelling with no latent heat, and the bottom panel shows the results with latent heat. In all cases, along the lateral walls where the airflow passes over the surfaces, there is additional cooling with latent heat model compared to the cases with no latent heat involved. The mucosal surface temperature are the coolest in these regions where the air flows over the surfaces. Temperature values were taken at cross-section slices. Two models performed similarly in the anterior half when the inlet temperature was highest. In the middle and posterior region, the inclusion of latent heat provided additional evaporative cooling effect at the air-mucus interface. The nasopharynx temperature was taken at slice Y7, and this value was used to compare with the measured data from Raouli et al. With the reference there cited at the bottom. We note that the model performed well when the latent heat model was applied, which provided the additional cooling effect, and the results at the nasopharynx were consistent with the measured data. The variation in the water vapor fraction, also known as the specific humidity, is depicted in this graph. The difference in the mass transfer between the two models slowly diverge, and the difference is most significant in the posterior region, where the cumulative effect of cooling reduces the capacity for the air to absorb the water vapor. This work proposed a Subwall model to account for the latent heat energy transfer during evaporation of water vapor from the mucus. The model recreated an adiabatic cooling process in a pipe successfully and performed well in 3 steady-state inhalation conditions evaluated against some experimental data, where the results demonstrated a cooler mucosal surface temperature when the latent heat exchange was included. A limitation of the current work was that it was applied to a single patient-specific nasal cavity and under a steady-state solution. Nevertheless, the Subwall model can be applied to other engineering applications that involve evaporation, such as water loss from swimming pools, human thermal conditions from perspiration, and evaporative coolers. The results in this presentation form part of an upcoming study titled Wet Surface Wall Model for Latent Heat Exchange During Evaporation, which is currently under review. It is anticipated that the pipe case used in this study and the UDF file will be available with the paper as supplementary material and will be made available at cfdresearch.com. This work is part of a larger set of projects held between colleagues at RMIT, Sydney and Auckland University and Westmead ENT. Thanks for attending this talk, and I look forward to answering your questions.